Hello and welcome everyone. You've reached the CyVerse Focus Forum webinar series. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar is on identifying transcription factor binding sites from ChIP-seq data, presented by Reetu Tuteja, a CyVerse science analyst based here at the University of Arizona. Like most of us, she's working out of her home for the time being. For those of you who are new to CyVerse, we are an NSF, which stands for National Science Foundation, funded cyber infrastructure project, and this free webinar series is designed to fulfill a key part of our mission to train scientists on how to use CyVerse's computational resources. First, let's quickly take care of some housekeeping before Reetu takes over the webinar. Today's presentation is roughly 30 minutes. We'll do Q&A at the end. Please open the Zoom chat window and type your questions there, and I'll read those for Reetu when she's done presenting. The slides and the webinar video recording will be posted to our website's webinar page later today for you to review at any time. Even before COVID-19 radically changed our world, CyVerse was working on tweaking our training materials and events to encourage remote online learning, not only to reach those like you who may not be able to travel or even afford to travel, but that many of our users find and use CyVerse when they reach a technical barrier or an educational milestone. So please watch our website for more information on those resources. And now I'm pleased to introduce Reetu, my colleague here at CyVerse. Reetu earned her master's in bioinformatics from Banasali University in India, and her PhD in plant sciences and bioinformatics from the National University of Ireland in Galway. Her research interests include the origination of new genetic material and its role in plant adaptations. And to partly explain why she fits in so well at CyVerse, she says she writes code for fun. So hello and welcome, Reetu.
Hi, Tina. Thanks for the introduction, and thanks everyone for joining in for today's webinar. Sorry, I have like a little bit of noise outside. Sorry about that. I'm missing a slide. Okay, so this webinar covers the basics of ChIP-seq assay and how to analyze ChIP-seq data. So we will cover a little bit about how to do the quality assessment for ChIP-seq data using ChIP-QC-R package. And we will be using CyVerse Discovery Environments apps to do analysis of a sample set. And for following the most part of the tutorial for this webinar, we don't require any command line expertise. So I wanted to start off by talking about a little bit about what ChIP-seq is and how this is used. So the questions around the center of a ChIP-seq experiment is, how do cells know what to do? So I'm taking humans as an example here. So human body has about like several trillions of cells, each carry the same genome, yet these cells do different functions. So what is it that makes these cells do different functions? So the function of a cell is largely depends on the gene expression. So is largely determined by the gene expression. So differences in the gene expression causes these cell types to have different proteins, and these proteins lead to the are responsible for the different functions of these cell types. So gene expression is regulated by the transcription factors, and which are not solely responsible for the gene expression regulation. Gene expression regulation in eukaryotes are also regulated at several different stages, from the accessibility of the DNA to the production of mRNA and protein and processing of the proteins. So these transcription factors are proteins that are responsible for the stimulation or repression of the transcription of target genes by binding to the regulatory elements. And the other factors could be like, for eukaryotes, could be the cell signaling, small interference, RNAs, and also the chromatin accessibility, RNA splicing. So both histone modification and transcription factor binding can be measured by the ChIP-seq assays. So a first step in the ChIP-seq assay is to treat the cells with the experimental condition that you desire, and the next step is the cross-linking step, where we treat with the formaldehyde to make the bond between the DNA and protein permanent, and the next step is the fragmentation step, where you separate the chromatin attached to the proteins. So normally the sonication step is used, sonication techniques are used here, and for the immunoprecipitation step, you treat the with the cells with a specific antibody for the protein to immunoprecipitate these proteins to pull them out of the solution, and from here we proceed for the sequencing, and the control data set for the ChIP-seq is input DNA or mock chip. So for the input DNA control, you treat them with a so there is a cross-linking step. We treat them with a formaldehyde, and also we do the fragmentation step, but there is no immunoprecipitation step. And for there is another control that you can use for ChIP-seq is mock chip, where you just use a non-specific antibody which doesn't generate any immunoprecipitated DNA. So this is how a typical ChIP-seq analysis workflow looks like. So we have the pre-alignment processing step and the alignment step, which is similar to any high-throughput sequencing data analysis. So basically, you measure the quality of your sequencing reads, quality process them, and do the alignments. So any standard DNA short-read alignment program can be used here, where you basically align your reads to a reference genome. And the next step is the peak calling step, where you're identifying the enriched regions that represents the binding site of your protein. And the last step is the post-alignment processing step, because the pre-alignment processing step doesn't measure the quality of your ChIP-seq, it's only for the quality of your sequencing reads. So we calculate here the cross-correlation score and FRIP score. So we will talk about these later during the presentation today. And for the demonstration part, I'm not covering the first two steps due to the time limitation, but I have put together a tutorial here where it has all the steps how to run ChipSeek using CyVerse Discovery Environment apps, and all the data set and everything is provided in this tutorial. So after you have the enriched represents that are likely to represent the binding site of your proteins, the obvious next step is to find your peak annotation and visualize your peaks into the genome browser, and do the motif discovery, which is to find a similar pattern of sequences in these enriched regions that define the motif binding site for these proteins. And if you have also the expression data for your experiment, that's useful to answer like which of the regulated genes in my expression data are likely to be direct transcription factors, direct targets for the transcription factor, and also to answer questions like if my transcription factor is an activator or a receptor. So some of you have expressed interest.
Reetu, excuse me, Reetu, your sound is off. Can you check on did your microphone turn? Okay, that's better. I can hear you now. I'm sorry to interrupt. Okay.
That's okay. It's because of, I think, the noise outside.
Got it. Okay, anyway, we lost you about 10 seconds ago. Thank you.
Okay, so I'm talking about the downstream analysis again. So after you have identified the set of enriched regions for that are likely to represent your binding sites of your transcription factor, the next obvious steps is to do motif discovery, which is to identify similar patterns of sequences inside in these enriched regions. So these define the binding site of your motif for your transcription factor, and other steps could be like to do the peak annotation, visualize the peaks in the genome browser, and also if you have expression data for your experiment, it's good to answer questions like which of the regulated genes in your expression data sets are likely to be the direct targets for your transcription factor, and also to answer questions like if your transcription factor is an activator or is a repressor. So some of you have expressed interest in covering the downstream analysis also, but this is out of the scope for today's webinar. So we will be covering that in another webinar that will plan out in near future. So I wanted to also talk about how the peaks are identified and how the quality assessment tools for ChipSeek works. So for a successful ChipSeek experiment, you see enrichment of the sequencing reads around the binding site where your protein of interest is binding. And a success of a ChipSeek also depends on the specificity of your antibody, and also how much enrichment you get in the affinitive precipitation step. So you see here the fragments here on the sense and the antisense strand, and the dark region represents the sequencing tags. This is on the 5' because of the nature of the short read sequencing. And this is how a typical ChipSeek signal looks like, and this distribution pattern is used by the ChipSeek peak identification programs to do the peak calling. So as a first step, these programs assess the distribution of the reads and then compare it with the background data set to check if the enriched regions are likely to be the real binding sites or not. And we can also use this information to check if the binding site, if your ChipSeek has worked or not. So you have like clusters of read on the positive strand and negative strand of ChipSeek. So the algorithm is to shift these reads base by base towards 3'N. So I'm going back to the previous slide for a minute. So if you calculate the correlation between the read count of the positive and the negative strand, you're likely to get a good correlation at the fragment length for a good chip signal, and you will get good correlation for the artifact signal here on the length of the reads. So that's how, if you calculate the cross correlation, which is basically the Pearson correlation values for the read counts for each base pair shift, you're likely to get two peaks here. So fragment length peaks are the real peaks, and the read length peaks are the artifact peaks. So this is how a successful versus a failed chip looks like. So for a successful chip, you are likely to get a higher peak at the fragment length, and for a marginal successful chip, you are likely to get a higher peak at the read length, but there are few good peaks also. And for a failed chip, you will just get the higher peak at the read length. So moving to the demonstration part now, user CyVerse Discovery Environment apps. So from this tutorial, the first step is the sample data set and pre-processing. And for the sample data set, I have selected a small, we're running a small E. coli data set for the FNR transcription factors, which is fumarate and nitrate reduction transcription factor that controls the expression of about 100 target genes in response to anoxia. And if you click on the discovery environment here, this is the page, this is the screen you will see after you log in, and you will see 3 tabs on the right, data, apps, and analysis. So data is to store, manage, and share your data, and apps is to discover, create, and use the scientific apps that are the tools that are integrated as apps in the discovery environment, and analysis is to find the status of your analysis and check the parameters that you have inputted. So if you click on data, you will see 2 folders here. So the first folder is your home folder, which is private, where the data is accessible private to you. And for the community data, this data is public data here. And if you click on the, I have provided all the example data set in this public folder, and the location of this data is provided here in the tutorial. And I have provided all the files for each step of this sample data set analysis. And if you click on ChipSeek webinar, you will see different folders for each step of the analysis. So sometimes this takes a while to come up. And for running the apps, so this is all the data, it's FASTQ files, alignment files, and then the quick calling files. So if you click on the apps here and search for MAX2, so MAX is model-based ChIP-seq analysis tool. So if you click on this app, you will see the by default analysis name is provided. You can change if you like. The output folder is provided. And if you click on the inputs, you can provide the treatment and the control files. So you browse through the database for this, and you'll find the data in the example folder here. So I'm providing the treatment files here, and then the control files. So you can provide the name of your experiment if you like, so it represents if any input is required. So I'm inputting SAM as my alignment file format. So SAM stands for the sequenced alignment map file format. And then you have to provide from MAX2 the size of your reference genome, which in our case is E. coli, and you have to provide that in the base pairs. So I will refer to my tutorial for this. So this is E. coli genome size is about 4.6 megabase pairs, and I'm going to leave rest of the parameters to default and click on launch analysis. So here in the notifications, you will receive the notifications if your analysis is submitted and it's running, and then it will give you a notification it's completed. So it takes about 4 minutes to complete the small data set. And then you click on the analysis tab here, and it will show you the status of your job that it is running. So since this is going to take about 4 minutes to run, I have some output files for MAX2 stored from a previous run for the same sample dataset. So maybe I can show you those. So for the MAX2 output, you get 3 output files by default. So the main file that we are going to check is the narrow peak output format. So for the transcription factors, the binding site is very narrow, but for the histone modification, this is like covered across the gene body of the the binding site is covered on the gene body. So those are sort of broad peaks. So for MAPS2, they provide a flag broad. So I'm clicking on this narrow peak file. So here you will get some information about the peaks, like how many number of peaks you have identified. The first column is the chromosome names, then the start of the peak, third column is end of the peak, and then you have the name of the peak, what is the score of each peak. So this score depends on if you have provided p-value as a cutoff or q-value, then strand, and this is the full enrichment compared to the background, p-value, q-value, and the center position of your peak. So we are going to use this analysis for this file for the next steps. Also, I wanted to mention that you can check from this file like how many number of peaks you have identified. For some reason, this analysis is failed. And I promise this works always. So you can go to output folder and you can check the logs, and that should give you an idea about why your analysis is failed. So you check in the error files here, and it should give you some idea what is the problem with your app or your analysis input. So this should be some sort of exception, listing outside of range. So maybe something has not worked well, the inputs are correct, the genome size is correct, all the parameters were correct. Okay, so I promise this works, so I'll just fix that and we'll put a note in the tutorial that what was the problem. You can download this narrow peak file here from the downloads, and I have already downloaded that on my local computer, and I will loaded this file and the alignment files into the genome browser, so we can maybe visualize some of the peak.
Reetu, again, your audio is mute. Okay, there you go.
Can you hear me now?
Now it's better. Yeah, it fades out. So keep going. Sorry.
Okay, I'm sorry about that. This is because we are working from home and CyVerse office might have been better for this. So the darkness of the peak depends on the score that you have got in your peak output file. And you can see like there are like some good peaks here, and this is the lighter one where you don't have a good score. So there is like, you don't have a good peak there. So I think I still have a couple of minutes here, so I can also maybe cover the chip quality assessment using our ChIP-seq package. This is the step 4 for this. So for this step, we will use the RStudio ChIP-seq app inside the discovery environment. So this app is a voice app, which is the visual interactive and computing environment, which is the latest feature of CyVerse Discovery Environment. So before this, all the apps inside the discovery environment were non-interactive, and for exploratory analysis tools, we can launch RStudio, Jupyter Notebooks, and any tool-based tools from the discovery environment. So again, I'm going to apps, and I'm searching for RStudio. ChIP-seq app here. So click on the app and launch RStudio from here. So again, you have to provide, you have a default analysis name. You can change if you like, and you have an output folder. And you can provide an input folder also. So if you provide any input data set here, then it's going to launch that inside your RStudio workspace. And then you click on launch the analysis, or just leave the default and use other methods to get the data in the RStudio. So I have already, I already have an instance running for this app. So here we are using the ChIP-seq package, and inside the, I'm providing my alignment files and max output, which is peaks file, to the ChIP-seq sample function. So after I have loaded all the sample data sets here, and I'm going to plot the FRIP score and the cross coverage score that I mentioned in the slides. So FRIP score is a fraction of reads inside the peaks. So ENCODE recommends that if you have a FRIP score for ChIP-seq, 5% means 5% of your total reads are inside your peaks, then it indicates a good quality ChIP-seq. So for this sample data set, we received about 10% of FRIP score, which indicates a good quality. And you can also plot the cross correlation scores for this data set. So mainly for each base pair shift, you have a cross correlation score for this data set. So this data set represents that you have a lot of peak, good correlation score at the fragment length. So this indicates that this data is of good quality. So that's all I wanted to cover for this webinar, and I'm happy to take any questions that you have.
Thank you, Reetu. It gives us some good time for some questions. There is one, and I'll read it. Thank you for the nice talk and demonstration of the Mac2 covers apps.
Thanks,
That's your comment there. So this person is wondering, does the Max2 app also do the plus VE and minus VE strand Pearson correlation, or is there another app that does that? Thank you again and best regards.
Okay, I think Max2 uses this information internally to build a model for identification of the distribution of peaks. But I'm not 100% sure if that gives this score somewhere. But that's definitely that you can calculate with the ChIP-seq package.
Okay. One of the questions I had, Reetu, was
yes.
It looks like you have everything there that you don't need to code. You don't need to know how to code, right?
Yes, definitely.
The ChIP-seq analysis. So other than understanding how to interpret log files, or if you are going to launch a Jupyter notebook or something, you might need a little bit of coding, but basically what you've shown here can do without any command line expertise.
So I would like to mention that for the last part, for the quality assessment using ChIP-seq, you need to be familiar with little bit of coding, but I have provided the full script here, and it's a very simple code. You're just putting the BAM files and the peak identified peaks file to providing to this function, and then you're just plotting. So it's very simple. Like you just have to copy and paste that.
Right. And that one of the things about CyVerse is down at the bottom far right corner, once you're in the DE, there is an in-app support chat there that we monitor during business hours. So, okay. Are there any other questions for Reetu? Okay, well, I'll thank Reetu so much for sharing her knowledge about ChIP-seq with us. And we'll be posting her slides and the video on our website webinar page later today. Please join us in 2 weeks on Friday, May 8th for the finale to our Spring 2020 Focus Forum webinar series. We'll be featuring Dr. Francesca Vitali, who will offer us a, quote, fruitful and fun introduction to data mining and machine learning using Orange, an open source visual programming software package for data viz, machine learning, data mining, and data analysis. So again, you can register for that by going to our website on our events and webinar page. And with that, thank you everyone. I'll see you in a couple weeks.
Thanks everyone.